technique can be used to detect genetic disorders in humans. Answer: polymerase chain reaction (PCR). Explanation: polymerase chain reaction (PCR) technique can be used to detect genetic disorders in humans. Polymerase chain reaction (PCR) is a technique that can amplify a target DNA segment. This amplified segment aids in the detection of various genetic disorder and further studies. It is a revolutionary method developed by Karai Mullis in the 1980s. Polymerase chain reaction (PCR) is based on using the ability of DNA polymerase to synthesize new strand of DNA complementary to the offered template strand. Because DNA polymerase can add a nucleotide only onto a pre-existing 3OH group, it needs a primer to which it can add the first nucleotide. Abhirami runs to her classroom as the school bell rings. What will be the probable rate of breathing in her lungs due to this vigorous act? Answer: 22-25 minutes per minute. Explanation: Abhirami runs to her classroom as the school bell rings. 20 to 25 minutes per times per minute is the probable rate of breathing in her lungs due to this vigorous act. As exercise intensifies and the body's need for fresh oxygen increases, the ventilation rate responds accordingly. The metabolic byproducts of exercise build up as a result of cellular respiration, and the amount of carbon dioxide in the system also increases to act as a buffer against these acidic byproducts. As the concentration of CO2 increases, the body responds by breathing deeper and more frequently to dispel the CO2. Working muscles also require fresh oxygen. The rate of respiration also increases to facilitate the delivery of oxygen to the bloodstream, where it is then transported to the working muscles. The rate at which cellular respiration takes place and hence oxygen is consumed varies with the general state of activity of the body. Vigorous exercise may increase by 20 to 25 times the demand of the tissues for oxygen. This increased demand is met by increasing the rate and depth of breathing. The carnivorous fish Gambusia, introduced in the lakes, ponds, etc., control a deadly disease in India, feeds on the larva of answer Anopheles. Explanation: the carnivorous fish Gambusia, introduced in the lakes, ponds, etc., control a deadly disease in India, feeds on the larvae of Anopheles. Gambusia elf Gambusia affinis, a native of coastal waters of United States from New Jersey southwards, introduced into India about 40 years ago from Italy and Thailand. These species can successfully survive in overcrowding conditions found in fresh water, brackish water, and salt marshes with high salinity. It feeds on aquatic and terrestrial insects. A single carnivorous fish that can consume mosquito larvae of Anopheles equal to its own weight each day, and hence known as mosquito fish. When subminimal stimulus is given, then answer muscles never contract. Explanation: when subminimal stimulus is given, then muscles can never contract. Muscles are excitatory tissue. Muscle contraction is initiated by a signal sent by the central nervous system CNS via a motor neuron. A motor neuron, along with the muscle fibers connected to its constituent, a motor unit. The junction between a motor neuron and the sarcolemma of the muscle fiber is called the neuromuscular junction or motor end plate. A neural signal reaching this junction releases a neurotransmitter, a choline, which generates an action potential in the cell sarcolemma. This spreads through the muscle fiber and causes the release of calcium ions into the sarcoplasm. The muscle fibers contract based on all or non philosophy, and the nervous stimulus should be above a critical threshold to cause muscular contraction. In case the stimulus is subminimal, muscles will not contract. This is called all or none law. Human cells in culture show a cell cycle to be completed in approximately 24 hours. Explanation: human cells in culture show a cell cycle to be completed in approximately 24 hours. Cell division is the process by which a cell divides to form two nearly equal daughter cells which resembles the parent. The cell cycle is the sequence of events which occur during the cell growth and cell division. It is completed into two steps: interphase and M-phase. In the cell cycle, interphase is the period between the end of one division to the beginning of next cell's division. It is known as the resting phase. In the case of human beings, it is around 24 hours. Then the metaphase, then the M phase takes place. A keystone species is removed from an ecosystem. This will not cause answer increase in the rate of growth of one or more populations. Explanation: a keystone species is removed from an ecosystem. This will not cause an increase in the rate of growth of one or more population. A keystone species is any plant or animal species that can control population and maintains ecosystem. Without keystone species, the ecosystem will be dramatically different or cease to exist along altogether. All species in an ecosystem or habitat rely on each other. Which of the following characterizes parthenogenesis? Answer: an egg develops without being fertilized. Explanation: an egg develops without being fertilized characterizes parthenogenesis. Parthenogenesis is a reproductive strategy that involves the development of a female gamete sex cell without fertilization. An egg produced parthenogenetically may be either haploid, that is, with one set of dissimilar chromosomes, or diploid with the paired set of chromosomes. Parthenogenesis has received considerable attention as a tool for the production of stem cells. The possibility of deriving stem cells from parthenogenetic embryos could eliminate the requirement to produce and destroy viable embryos and may reduce the ethical concerns surrounding stem cells research. Biosystematics aims at answer the classification of organisms based on their evolutionary history and establishing their phylogeny on the totality of various parameters from all fields of studies. Explanation. Biosystematics aims at the classification of organisms based on their evolutionary history and establishing their phylogeny on the totality of various parameters from all fields of studies. Camp and Gilly in the year 1943 coined the term biosystematics. In simple, systematics equals to taxonomy plus evolutionary phylogenetic relationships. Morphological, cytological characters are alone are not criteria for distinguishing and classifying organisms from one another. One has to take into consideration the characteristics and differences from other disciplines of science such as cytology, genetics, physiology, ecology, molecular biology, numerical taxonomy, and any other available sources for classification. Biosystematics is a branch of systematics concerned with variations and evolution of taxa.